Ribosome. Ribosomes are tiny spheroidal dense particles having about 150 to 200 angstrom diameter and contains equal amount of RNA and proteins. Ribosomes are found in all cells. Ribosomes are found either in free state in the cytosol or attached to endoplasmic reticulum. Ribosomes have sedimentation coefficient of about 80s and are composed of two subunits, namely 40s and 60s. See here in this figure, you can see here it is nucleolus, then nucleus, and endoplasmic reticulum. So rough endoplasmic reticulum shows the attachment of ribosomes. See here these dots, they represent ribosomes. So these ribosomes, which are concerned with protein synthesis or sites of protein synthesis, so we will discuss about them here. So, ATS type of ribosome is composed of two subunits, 40S and 60S. So, see here in this figure or sketch, this is smaller subunit. And larger subunit actually, and this is smaller subunit. See this one, this is smaller subunit, while this one is larger subunit. So this larger subunit, it is 60S, and this smaller subunit, it is 40S, and overall this type of ribosome is 80S type of ribosome. So in eukaryotic cells, this type of ribosome is seen, while in case of prokaryotic cells, 70S type of ribosome one can observe. Here smaller 40S ribosomal subunit, that is this one, so it is ellipsoidal in shape and consists of one molecule of 18S ribosomal RNA and 30 proteins. So these proteins, they are named as S1, S2, S3 and like that. So this is the case of smaller 40S ribosomal subunit Its shape is ellipsoidal and it consists of one molecule of 18S rRNA. 18S rRNA and 30 different proteins which are named as S1, S2, S3, like that. Now 60S, that is this one, larger subunit. It is round in shape and contains channel. See this channel, you can see here, contains channel through which polypeptide chain makes its exist. So this 60S larger subunit consists of three types of rRNA molecules. .288 rRNA is one, second 5.8 rRNA, and third 5S rRNA. So these three types of rRNA molecules are there in this larger subunit, and 40 different types of proteins. 40 different types of proteins which are named as L1, L2, L3 and like that. So we will see some of the details related with the ultrastructure of ribosome. Ribosomes have no unit membrane, any unit membrane. It measures about 170 to 230 angstrom and its sedimentation coefficient is either 70S or 80S. Each ribosome is distinguishable into two unequal subunits. Here we have seen one larger and second smaller subunit. Larger subunit is having three chemical sites. This larger subunit has three chemical sites. One is decoding site, second is condensing site, and third is exit site. So three sites are there on this larger subunit. About 75% ribosomes of cell remain attached to endoplasmic reticulum membrane, and this attachment is through glycoprotein named as ribofourin. A 0.001 molar concentration of magnesium molar concentration of magnesium, if it is there, then ribosomal subunits gets united to form a dimer. A tenfold increase of magnesium ion concentration is there then ribosomes forms a dimer. Seventy-S ribosomes form 100S dimer. 70S ribosomes form 70S ribosome forms 100S dimer when there is tenfold increase in magnesium ion concentration. And if the magnesium ion concentration becomes less, then dissociation of these dimer take place. ATS ribosomes, they forms 120S dimer. If concentration of magnesium is decreased by tenfold, the ribosomes dissociate. 70S ribosomes dissociates into 50S and 30S. 50S and 30S. And ATS into 60S and 40S subunits. These subunits are further dissociated into RNA and protein components like this. Only unemployed ribosomes dissociate in low magnesium ion concentration and not those which are engaged in protein synthesis. Engaged in protein synthesis, call them as active ribosomes. And those which are not involved in protein synthesis, call them as unemployed ribosomes. Stoffler and Whitman in year 1977 proposed model for ribosome, quasi-symmetrical model for ribosome, and it is here. This model is represented here. This model differentiates ribosome into base, head, and body. head and body components are separated by dent and cleft. Dent and cleft. The base has central protuberance. Base has this one, central protuberance, and two arms. This is one arm and this is second arm. A tunnel is enclosed between two subunits. See, this is tunnel. In between these two subunits, this tunnel is enclosed. So this is what the model given by Stoffler and Whitman. Now chemical composition, if we see of ribosome, 70S ribosome contains RNA and protein in 2:1 ratio, while 80S ribosome contains RNA and protein in equal amount, that is 1:1 ratio. Molecular weight of 80S ribosome is about or in between 4 to 5 million, and molecular weight of 70S ribosome is 3 million, and it is measured in Daltons. Major function of ribosome is a site of protein synthesis. Means we can say ribosomes are concerned with protein synthesis.